We're back.
And we're back. All right.
Okay, great.
Turned on here. All
Oh my
right.
gosh. Okay, hi.
Hi.
Just been
Thanks everyone for joining us tonight. My name's Catherine. I'm with the MSU Science Festival, and we are here.
We're back.
And we're back. All right.
Okay, great.
Echo here. Let me just mute myself. Sorry about that. All right. All right. I'm Catherine from the MSU Science Festival again for another night of Science After Dark. Tonight we are joined by Tess Brzezinski, the founder of Fungi Freights from Detroit. Welcome.
Hi, thank you so much. I'm really happy to be here.
Great. Well, glad to have you. Before we jump into tonight's presentation, could you introduce yourself and tell us a little bit about what Fungi Freights is?
Yeah, absolutely. So hi, everyone. Thanks for joining. My name is Tess. I am a profession an environmental scientist. I work for a civil engineering company, but I have been involved in studying fungi for over 10 years. So it's a big part of my life. And I started Fungi Freights, basically equipping a freight container to be a lab space for people to engage in the mycological realm and providing a space for citizen science and for people to get that experience with fungi and mushrooms. So
that's incredible. So tell us a little bit about what you're going to be talking about tonight.
So yeah, we're gonna basically be talking about mycelium, which is the vegetative structure of fungi. What we primarily don't see. We usually, when we think of a mushroom, we think of the fruiting body that pops up from the ground after a few days rain. But we're going to talk about the vegetative structure and everything that goes on underneath. We'll give a little bit overlay of fungi and mushrooms as well, but primarily of mycelium.
Awesome. That sounds great. Well, are you ready to get started?
Sure. Yeah, let's jump into it.
All right.
So
while we get set up, if anyone has any questions throughout the live stream, feel free to enter them into the YouTube chat, and we will be sure to cover them in the Q&A session following.
All right, let me just share my screen here. Oh, shoot. Sorry. Okay, here we go. Looks good?
Yep, looks good.
Okay, so welcome everybody, and let's dive into the realm of fungi. So we're gonna go back to roughly 420 million years ago to the land of the Prototaxides. So the Prototaxides fossil is thought to be the oldest multicellular organism on the planet. So scientists discovered its fossil in 1859, and it was thought to be like, these are about 20 feet tall, by the way. They were really large organisms dominating the earth, and they were thought to be a conifer-like tree or an algae. But after doing further studies, I believe in the 1950s, it was discovered through isotopic dating and carbon nitrogen ratios that this organism was not obtaining carbon from the atmosphere, actually from an organic source. So it was actually feeding off of carbon from the earth. So then removing it from being a plant to actually a fungus. And we'll talk about how plants or fungi obtain their food in a little bit. So these multicellular fungi actually paved way for terrestrial life on Earth, because there was no soil at one point. The Earth's crust was very hard and rigid, and these organisms with the mycelial structure actually broke down that really hard crust of the Earth and thus created soil, paving the way for terrestrial life to begin on Earth. So that's kind of the beginning, and here's the actual fossil. I believe this is Saudi Arabia. It's from Smithsonian Magazine here. So we'll talk a little bit about phylogeny. The phylogenetic tree is an organization of all life on Earth in a hierarchical form. This is just a portion of it. pointing out eukaryotes. So eukaryotic organisms are multicellular, and they have nucleus with specialized organelles. So at one point, fungi were placed in the same kingdom as plants. And after studying further their characteristics and how different they are from fauna and flora, they were actually put into their own kingdom. And we'll talk about these characteristics in the next slide. But the really cool thing is now they're actually in a supergroup with Animalia. So it's called Opisthoconta. And this supergroup actually contains fungi and Animalia. And that's how close we are ancestrally with fungi. So I think that's a super cool discovery. And it just reminds us that, maybe we do come from fungi, or we have been coexisting with this organism for a very long time. So some of these characteristics that fungi have that do place them in a different category than plants are the fact that they're eukaryotic. So, I mean, plants are too, but they're eukaryotic organisms. They're multicellular and they're non-photosynthetic. So unlike plants, they didn't develop a thing called photosynthesis where you can just like bathe in the sun and create your own food. Unfortunately, fungi cannot do that. So they're heterotrophs, just like we are. They have to seek out their food from a external source. They undergo extracellular digestion. So they actually digest their food outside of themselves. And we'll talk about that in a little bit. Their cell wall is comprised primarily of chitin. So plants, their cell wall is cellulose, whereas fungi, their cell wall is chitin. And chitin is a polymer of what like arthropods and crustaceans are comprised of. So that's why fungi has that like really different texture than plants. Sometimes they're squishy, sometimes they're more rigidy and hard. So it's because of the chitin in their cell wall. Most of them are non-motile. I'm not going to say that they're all not, because some actually do contain flagella and they can swim. They intake oxygen and exhale CO2. So once again, this is the opposite of plants and similar to us. We intake oxygen, exhale CO2. Likewise for fungi. They're decomposers. They can undergo asexual and sexual reproduction, and they produce spores and not seeds. And as you can see, they're very diverse. I took all these photos up at the University of Michigan Biostation up in Mayo, northern Michigan, Mayo, Michigan. So even right here in Michigan, you can see the very broad diversity we have of fungi. So really quick, we'll just talk about the 7 phylums. I call it the queendom fungi or fungi. So classification within like the fungal queendom is always under dispute and changing, but for now, these are the 7 phylums. And the first one we have is Chytridiomycota. These are some of the oldest fungi. They're very simplistic, and they produce zoospores, which cause them to be motile. So they can actually, they're usually found in aquatic environments. Microsporidia, these are also a simple, really simple form of fungi, and they're mainly parasitic, thriving in like insects, human, and other animal hosts. Then we have Glomeromycota. I'm going to go on a little rant about this phylum here. It's one of my favorites. Super cool, dating back to like 450 million years ago. This like genus of fungi is super interesting. Not only the fact that they produce glomalin, which is a protein that is really essential to soil health. It actually like binds soil together and gives it, helps it retain moisture, and just is really helpful for the microbiome within the soil. But they were pretty much the first or oldest terrestrial fungi on the planet. And the cool thing about them is that their spore size is significantly larger than a regular spore. So a regular spore which is like the seed of a fungi, usually can get up to about like 20 micrometers, whereas a glomeromycota spore can reach up to about 800 micrometers. So you can kind of see the drastic difference in spore size. And the really, really cool thing about them is that they can contain DNA from up to 35,000 different varieties of DNA, some from other phylums. So like a regular spore would just contain that specific genetic material of that fungi. Our cells just contain DNA specific to like who we are. Glomeromycota is pretty much the mothership of like genetic variation in fungi. It can contain 35,000 different genetically different DNA. So it's really bizarre, but a way to think of it is like if we could have DNA of like a shark or a cat or an alligator. So basically it's the mothership, and it just like contains a vast variety of DNA. And I don't know if this was some form of like survival to like spread DNA, you know, because spores can spread like from continent to continent. Either way, it's a really interesting fact. And if you want to learn more about it, you can look up glomeromycota and DNA. Okay, so moving on, endovrant. We have the blastocladiomycota. These are saprobes. They break down dead organic matter. The neocalammastigomycota. These are anaerobic fungi, mainly dwelling in environments with like little to no oxygen. Then we have the ascomites. These are sac fungi, and the basidiomycota, club fungi, the most common fungi or mushroom that the layman usually thinks of, just a guild mushroom. So, okay, moving on. Really quick, just to clarify and distinguish when we use the word fungi and when we use the word mushrooms. So how I like to break it down is that all mushrooms are fungi, but not all fungi are mushrooms. So a mushroom is the fruiting body of the fungi, and not all fungi produce fruiting bodies. So the fruiting body is pretty much like an apple to a tree. It's the reproductive structure that its sole purpose is to reproduce. And they do this via spores. So they'll release their spores and then decay, and the cycle will go on. So fungi like molds and yeast, most of them do not produce fruiting bodies. So we wouldn't call them a mushroom. Okay, to tap into reproduction a little bit, just to show you or explain a little bit how mycelium is formed. So like we were saying, mushrooms or fungi can reproduce asexually or sexually. So they undergo mitosis and meiosis. So in asexual reproduction, they undergo mitosis, and these spores are going to be haploid, meaning that they only contain one set of like genetic material. So they're going to be completely identical to the parent. So this doesn't really offer much like genetic variation. And I think maybe sexual reproduction developed in fungi to get that genetic variation and allow them to survive better. And it as like adaptation is easier when you have a broader array of genetic material. So in sexual reproduction, a lot more fun, cool stuff going on. So very similar to our reproductive cycle, they undergo meiosis. The spores will be diploid, meaning that they will contain both genetic material of each fungus that like came together. So the cool thing I like to bring up is that mycelium contains mycopheromones. So as they're just like hanging out underground, wiggling around together, if they are like compatible and like, oh, I like your mycopheromones, they'll fuse, and then that's where the reproductive structure will derive from. So it's pretty interesting that they give off those chemical signals like we do. So this is just a reiteration of the same information, but in illustrative form, just to give you a better perspective. So this is a Basidiomyces, a regular, just common guild mushroom. So the spores will release, and if conditions are right, meaning the environment has to have like a consistent moisture level, pretty regulative like temperatures, and a good nutrient source. So not all spores will actually germinate. Some get eaten by insects or animals and will join into the food web. But for those that do germinate, they will produce hyphae, and we're going to talk about hyphae. So hyphae is a beginning form of mycelium. It's just a single protoplasm, and once it branches, it becomes mycelium, and that mycelium will give rise to primordia and to a mature mushroom, and then the cycle repeats. Just to dive a little bit into hyphae, so they're long tubular filaments. Some are septate, some are aseptate, just meaning that some have a septum that separates the nucleus and the organelles from one another. We won't get too much into that, but so here's like the cell wall, and beneath the cell wall is a semi-permeable plasma membrane. And this allows the inflow and outflow of chemicals from the environment. And we'll talk about how that's important for digestion. And here's just a realistic view at 1,000 times magnified of hyphae. So really quick on hyphal growth, because it's super interesting. It undergoes apical extension. So it grows from the tip. And it's a German word known as Spitzenkörper that means, it explains this whole phenomenon of these vesicles here at the tip. They're unknown compounds. It's still kind of a mystery of how this growth occurs. So I'm like, okay, so they're growers, not showers. It's just like not really showing us what's going on here. It's just like, here are these vesicles that spit out and will digest as part of the cell wall and continue on growth. So pretty interesting that it's not completely known yet. So it could be something that will spark someone's interest to kind of study. And here's a little clip just to show you how it actually is growing out from the tip. Interesting. Okay, so when hyphae fuse or grow together and branch out, that is what we know as mycelium. Mycelium is very tenacious. Although being one cell wall thick, it's super durable. It's super hardy. It really is responsible for holding the forest floor together. So the fact of it being seeming so stringy and thin, it actually is really powerful. So it grows out in like the pathway of its food source. So as you can see here on the left, growing in a agar petri dish, the nutrients are pretty well dispersed. So it'll actually grow in a circular formation, trying to digest all the nutrients within its pathway. So it's very sufficient in its like growth patterns. Over here on the right, you can see that it's going out pretty laterally, but it's still trying to branch out and get the nutrients like within its pathway. So it's super efficient in its growth patterns. So here we'll get on to the digestion portion. So fungi undergo extracellular digestion. So they actually secrete enzymes outside of themselves from the hyphal tip into the environment, and digestion like goes on outside. So it's like if we were to just like throw up like on our food and then just like lay on top of it and reabsorb it. It's really bizarre because they don't have to reabsorb the nutrients from where the enzymes were excreted. So that's pretty sufficient as well, because they can secrete enzymes and then keep growing out. And then this area over here can like come by and absorb the nutrients. So pretty interesting, and just shows kind of how different, they have an external stomach pretty much. So they break down the polymers, enzymes break them down into like simpler structures to monomers, and then they'll get reabsorbed. So fungi obtain their nutrients in 3 different ways. The first one we're going to talk about is parasitic fungi. So basically they obtain their nutrients from a living host. So they will survive off a living host, eventually killing it off. So we have Ophiocordyceps unilateralis. This is super interesting. It's an insect pathogen. So it specifically utilizes carpenter ants for a host, and these carpenter ants will like eat these leaves that have the spores of the Ophiocordyceps on it. Therefore, the spores will enter its digestive system where it'll germinate. And the mycelium, it was thought that the mycelium was taking over its central nervous system and like causing it to become zombie-like. But newer studies are actually talking about the mycelium taking over each organ and actually moving the organs from within. So what will happen is this ant will become like a zombie, and just it'll only know how to walk upwards. So basically it walks all the way up to the top of the plant and then plunders to its death. So it pretty much causes the ant to become like a suicidal zombie. So this is really cool. There's some videos on it and more information if you want to look up carpenter ants and Ophiocordyceps. To the right, we have Armillaria, which is a honey fungus. It's actually a pretty good edible. This is parasitic to furs. It's actually the largest organism on the planet. So if anyone asks you like, oh, what's the largest organism on the planet? No, it's not a blue whale. You can be like, oh, it's a fungus. I guarantee someone will be pretty proud or actually surprised that you know that. So it's not the mushroom itself, it's the underground mycelial mat. And how this was discovered is that arborists were studying in the Blue Mountains of Oregon, and they were noticing all of this like tree death. So they were taking like root samples from like 100 different trees, and they noticed that majority of them were infected with the honey fungus. Then placing it together, doing further studies, and they traced the genetic material, like the mycelium, all the way back to like a single genetic material. So the entire mycelial mat is genetically identical. So it's about 35,000 tons in weight, spanning 2.4 miles or like 2,200 acres, just to like give you perspective of how like massive this mycelial mat is. So although parasitic, it's still pretty cool. That's the largest organism on the planet. Then we have like my favorite, mutualistic fungi. This is a lot nicer relationship. So on the right, we have lichen. I love lichen, so beautiful. It's actually a symbiotic relationship of fungi and algae. So they survive together, sharing different nutrients amongst each other. They're very slow growing, so it's okay to collect lichen if you see it like falling on the ground, like from fallen bark. But usually it's not good to harvest it just because it does take a long time to grow, and it's pretty precious. So over here on the left, we have a mycorrhizal symbiotic relationship. So This mutualistic relationship occurs in about 90% of all plants and trees. It's super beneficial. The mycelium will actually like fuse with the plant or tree roots and expand like the whole rhizosphere. So in exchange for like sugars and carbohydrates from the plant roots, the mycelium will exchange minerals and different nutrients. Mostly, the most beneficial thing I think is that mycelium can go to further depths of the earth. It can actually go as far as the calcareous bedrock, and it can bring up that phosphorus, that calcium, different minerals that tree or plant roots would otherwise be unable to retrieve. So that's pretty cool stuff. So they create this underground network. They kind of like connect, you know, underground life and provide this like form of communication. So a mycorrhiza is the area where the plant and the tree, or the plant and the mycelium will kind of fuse together. And then it will kind of go on. But through that, it creates this like beautiful communicational network, allows things to share nutrients and just like share, I don't know, they're probably like talking, hanging out under there, I don't know, but it just basically expands like this whole rhizosphere, and allows plants to cover about 700 times more like biomass of soil than they would otherwise. So how this interacts in the environment, kind of is what we were saying. It's really good at like retaining moisture in soil and exchanging nutrients and holding the forest floor together. It really provides a really strong root system. And this picture over here on the right, I just had to add it in there because I find it funny. A professor in college, I was taking a terrestrial ecology class, and he just totally was weirded out and didn't really like the fact that I was so obsessed with mushrooms. And he really did not like mushrooms and kind of would write me off every time I would talk about them or bring them up. And I was like, whoa, dude, you're a terrestrial ecologist. I don't understand how this makes sense. But part of our field exams were to dig these soil soil profiles. So we would dig them and we would come back and kind of study the layers of the soil. And we came back about a day or two, and this like beautiful cluster of mushrooms just was like coming out of our like soil profile. And I was just like, aha, there they are. And I don't know, just found it kind of enlightening. So on to mycorrhizae soil, just showing here again a tree or plant root and its rhizosphere. So if you see this area within the soil that does not contain mycorrhizal mycelium, it doesn't have as many out branches to obtain nutrients. Whereas over here, when there is presence of mycorrhizae, you can see how further expanded its ability is to retrieve nutrients back to the actual root. And this is just the different organisms that are involved in that rhizosphere. Okay, and the third form of nutrient uptake and how fungi uptake their nutrients is saprophytically. So saprophytic fungi are awesome, and I like give a huge shout out to them because they're responsible for breaking down dead organic matter. So although it's like, ew, like look in your fridge and there's like some moldy food, it's actually a super amazing transition. It's breaking down those nutrients that would otherwise be locked up and inaccessible to other biota, and actually breaking it down and allowing those nutrients to get filtered back into the cycle. And the really interesting thing about saprophytic fungi, well, they're pretty much responsible for 90% of all decomposition. And they're the only thing on the planet that can break down lignin. So we're going to talk about lignin. But lignin is what it's a polymer that it helps make up woody plants and trees. So it's a component within, it's a really complex component that's in the secondary cell wall of all woody trees and shrubs, and it gives it that woody structure. So without saprophytic fungi, any tree that falls in the forest, any twig or stump would not get broken down. So we would just be like miles high in like dead organic matter of like trees and woody shrubs. So yeah, just a little bit more on decomposition and nutrient cycling. Like I was saying, 90% responsible for 90% of all decomp. They cycle the nutrients back into the cycle for other biota to be able to uptake them. So it's really, really a beautiful process. It's kind of like gives death life again. So we're going to move on to mycelium for environmental cleanup, and why I'm bringing this up right after the saprophytes is because of the way that mycelium can break down certain pollutants in the environment is through saprophytic fungi. And we'll get into that. So we know that, pollution is an ongoing problem. It's been on the rise since the Industrial Revolution. CO2 levels are rising, which is like an abundant greenhouse gas, and it's having a major effect on our climate, thus affecting a lot of biota. And I think it can be very, lead to like detrimental consequences if it hasn't already. So we're going to get into micromediation. We're just going to tap into it because it's a whole talk on its own. So just to give you a very broad introduction, mycelium has the ability to break down certain pollutants in soil or waterways, primarily hydrocarbons, because they are like carbon and hydrogen based. So they do this with their extracellular enzyme system. And the same way that, the hyphae will secrete the enzymes to break down food in its environment, it can also do this with certain pollutants and break them down. And we'll talk about why and how this can actually happen. So going back to lignin, I'm going to talk about polycyclic aromatic hydrocarbons, or PAHs. These are chemicals that like naturally occur in crude oil or coal. So when these fossil fuels are burned, they release these chemicals, and once they rise into the air and dissipate back down, it can be a problem with contaminating soil. So if you see here the comparison on a molecular structure to PAHs and lignin, they're both aromatic. So they share that structure of an aromatic ring structure. So like I was saying, saprophytic mycelium contain a lignin enzyme system, so they can break down lignin. So if they can break down lignin, it's like, oh, hey, here's a really similar structure. Like, let's break that down too. Like, that's how cool they are. They will just like, sure, we'll eat you as well and utilize you and break you down. So that's just like kind of an overlay of how this actually can occur. We've done some like applied soil mycology practices with micromediation. So micromediation was actually, it was discovered by Paul Stamets. However, he didn't put a patent on it because his theory is like everyone should be able to do this and utilize this. So it's been studied like in tabletop, like on a tabletop like version. People have done like some applied micromediation. We haven't really seen it like work on a very large scale. And I don't think that's how it's meant to be. I think it's meant to be implemented in small areas all around, therefore creating a big, large area. So there is ways to do this. And I do give a talk solely on micromediation. I do give workshops on it. So if you're interested in this, you know, we can chat later, shoot me an email and see how you can actually get out there and do some applied micromediation in your own area. So we're getting near the end here, but this is super, super good stuff here. And I really want to share it with everyone. If you haven't already heard of mycelium and how it's actually helping recolonize the honeybees. So for, I don't know, quite some time, bee colonies are facing like a major collapse. It's super devastating. There's many reasons for this, you know, pesticides, loss of habitat, monoculture or monocrops. That's just kind of destroying the vitality of honeybees. And they're super important to our vitality. They pollinate billions of dollars worth of crops a year. They're responsible for about 30% of our food supply. So without these pollinators, we could be facing like a kind of a really crazy collapse within like food security and just like our own mentality as well. So we're going to get into the varroa mite. So I think many of you are probably familiar with Paul Stamets, and he discovered something amazing, linking the mycelium to the honeybee. So really quick little background on the varroa mite. This is responsible for so many colony collapse, so many honeybee colonies to collapse. So it's native to Asia. And once it was introduced here, it kind of started taking over. And what it does is it will infect like the larva or like a baby bee, and it kind of just attaches onto like its back, and it will just feed off of the blood. And this is super detrimental. And a lot of things have been, a lot of things have been implemented to try to like ward off this varroa mite, but nothing has seemed to work. So Paul Stamets kind of put this whole thing together that he saw his honeybees kind of feeding on his outdoor mycelia, outdoor like mushroom garden beds and mycelium there. And, after like studying years later, he came up with a vaccine for the varroa mite, which is a combination of the reishi, amadou, and the chaga mushroom. These are really amazing medicinal mushrooms. They've been used for thousands of years and have many immunological benefits. So bees have an immune system as well. So basically putting the serum together and allowing bees to feed off of it, it's shown great results in warding off the varroa mite. So that's also a whole other topic, but that's just also more of how mycelium is so magical that's actually saving the honeybee, which is super amazing. Okay, and if you want to look that up, Paul Stamets, his research is published in Nature, one of the oldest like scientific journals, and it's a really good read. Okay, getting into mycelium for sustainable biomaterials. So not only are they just like saving everything, they're also being made into compostable, sustainable biomaterials. So that's me just like nerding out at a mushroom conference, playing with these micromaterials. So they can, for one, replace styrofoam, which styrofoam just completely sucks. It's horrible. It's a terrible thing. It's a single use. I mean, how many times like have you seen like somebody in the office just like grab a styrofoam cup of water and like throw it away and then like 10 minutes later go get another one? It's just like this terrible product that can even be toxic. It's non-biodegradable. It persists in the environment. It's so lightweight that even recycling it, or like when it's in the landfill, it often like blows away and like gets into the other habitats and environments. The production process is the 5th largest generator of hazardous waste. It actually comes from crude oil. So they're burning like fossil fuels at like a enormous rate to produce styrofoam. Terrible. So an awesome alternative is something known as mycofoam. So I actually was like working with mycelium years back, and like when I was first starting to grow it, I was like, wow, this stuff is like super cool. It like takes over whatever substrate it grows in. I'm like, this would be a really cool like sponge or something, like thinking of like ways to utilize it. And then I was looking into it, and then I find Ecovative Design, and it's a company based out of Green Island, New York. And they were already doing it. And I'm like, so funny. But they are amazing, and they're making these myco materials to replace styrofoam packaging. So how the production process works, all that it entails pretty much is agricultural waste, water, and heat. So no burning of fossil fuels, no like non-sustainable or non-resourceable resources. So how this actually works is you'll obtain agricultural waste, which is great because it is allowing you to reduce waste in your community. And you basically grow the mycelium in a mold, and then you'll heat it, and out pops this product. So basically you heat it to stop the growth process, and to kind of heat treat it to keep it in its like in a hardy form. So these materials are super cool. They've undergone like heat analysis, tensile strength, and like tests to show that they are high performance. They are insulators, fireproof, and like waterproof, and they're biodegradable. So like once you're done with it, or you get your package, you can honestly just like throw it out in your compost or your garden. And IKEA actually has just I don't know, the last 2 years or so, they actually adopted using mycofoam, which is so cool. So yeah, go IKEA. Sweden's on top of things. Also being used for like building blocks. As you can see, here's a very nice chair here made solely of mycelium, and even starting to like play around with like building things, like even structures with mycelial blocks. So if you want to learn more, I would maybe look up Ecovative Design or just myco materials. Even producing a mycelium like leather. As you can see, it's like super durable. It looks a lot like leather. This is a company known as Bolt Threads, and they call their mycelium leather Milo. Here's a cool purse. Gotta get you one of those. So yeah, if you're interested, Bolt Threads is kind of on this and creating myco leather. And last but not least, I'm going to talk about mycelium for personal well-being, and just talk about 2 of my favorite mushrooms. This here is Ericium erinaceus. It's the lion mane mushroom. I took this picture many years ago in northern Michigan. As you can see how its structure is like super like eye captivating. It's beautiful with this like icicle tooth like structure. It's actually super delicious. It tastes like lobster, like when cooked up in butter. And the mycelium has more beneficial properties than actually the fruiting body, but the mycelium contains medicinal properties that actually induce neurological growth and repair. So it's really great for cognitive function and supporting the nervous system. So it can actually repair neurons that have been damaged and help to induce neurological growth. So it's a super awesome fungi. I think we all should be taking it as a supplement. We've been seeing studies where it's helping with dementia. And like, I think dementia is super sad because it's like people that we look up to for like history. You know, our elders are like losing their memory and it's harder for us to learn from them. So start giving this to like your loved ones, your mom and dad or grandma, grandpa, whoever. And then I want to talk about Psilocybe cubensis. So psychoactive mushroom, one of my favorite mushrooms. I think they're amazing. They're finally being studied by like John Hopkins and Harvard for treating like PTSD, addiction, depression, and even like the fear of death of people that are like dying of like certain diseases and are scared to die. They've been undergoing these treatments by taking psilocybin and undergoing that kind of experience, psychedelic experience, and there's a lot of benefits from it. So just to explain a little bit, the psychoactive property is psilocybin, but once we ingest the psilocybin, our body breaks it down into psilocin. And that actually attaches to our serotonin receptor because, as you can see, the molecular structure is super similar to serotonin. It just differs by a phosphate group. So it's actually just like enhancing our, like, it's like flooding our receptors with serotonin instead of depleting it. So super good stuff. And I think with microdosing on with psilocybin and lion's mane together, it's probably super like one of the most beneficial things for cognitive function and opening up new pathways in our brain. So that's pretty much all I have for you. So an overview is just basically mycelium is ancient, it's powerful, it's tenacious. They're really great adapters. They play like a super important role in soil dynamics, connecting the webs of life. It allows people to kind of dabble in mycology and citizen science. They're kind of cleaning up after us, all the damage that we have occurred here on Earth and the life upon it. It kind of just can, you know, clean it up for us, which is something to be super thankful for. And it helps sustain all life on Earth. And that's just a quote that I wanted to end with by John Burroughs. I had to add him in here. I mean, look how cool this guy is. He probably loved mushrooms. He just looks like a mushroom guy. So it is well to stop our stargazing occasionally and consider the ground under our feet. Maybe it is celestial too. Maybe this brown, sun-stained, sin-stained earth is a sister to the morning and evening star. If it should turn out to be so, it seems we have many things to learn over again. So yeah, I just think that is it resonates really well with me and like the whole with mycelium as well. So thank you, everybody. Here's just some information on myself and Fungi Freights. If you want to learn more, you can go to my website, kind of learn more about what I'm doing here in Detroit and surrounding areas. So thank you so much.
Great. Thanks, Tess. That was wonderful. Really insightful. We can enter our Q&A section of the presentation tonight. If anyone has any questions, feel free to enter them into the YouTube chat. We've gotten a couple questions so far, so we can jump into those. Earlier, Scott was curious about some of the fungi found at Chernobyl. Do you know?
Yeah. So there is fungi that is surviving off of radioactive material. I'm really glad that you brought that up. That's it's super cool stuff. I haven't really researched much into it, but we know that there is fungus growing like in the walls in Chernobyl and feeding off. They're actually utilizing radioactive material as a food source. So they're breaking down radioactive material, which I think is also, you know, a form of how they are so adaptive and maybe how ancient they are, to be breaking down radioactive material. I mean, they're just so adaptive in the earth already, like with uranium and such. That's a good point. I don't really have too much information on that, but we do know that there is fungi growing and feeding off of radioactive material.
Very cool. From one of our younger viewers, we had a question. Do you know where the varroa mite comes from? Sort of how this became an issue?
I do know it's native to Asia. I'm not sure exactly how it migrated here, but it is native to Asia.
That's a great question.
That is a good question.
Another question we got, could you talk a little bit about mushrooms that you can find out in Michigan, just walking around? And we also had another question, are they only found on dead trees?
Okay, yeah, good question. So we do have a broad array of fungi. It depends like what you're looking for. If you're, for edible fungi, we do have the lion's mane. We have the morel, super renowned here in Michigan, the delicious delicacy. The season's actually like just starting. It'll probably go through May, so make sure to look out for your morels. I was gonna, I had a talk set up on morels, all on like a beginner's guide to morel hunting. But unfortunately with the coronavirus, I wasn't unable to give that, but you want to look in like disturbed areas and like by roadsides and dead elm trees. So that's a really good mushroom to look out for. We have the chanterelles. They come up in July. That's another great edible. They don't always grow on dead trees. So when we were talking about the different forms of how fungi obtain their food source, so if it's a saprophytic mushroom, it will grow in dead or organic matter. So that you'll see them growing on like dead fallen trees. Some that are mycorrhizal, like a morel, will pop up from the ground because it's actually having a symbiotic relationship underground. Some are specific to like specific trees. They like certain trees and will have that relationship with a certain tree. So it's good to know your trees when you're mushroom hunting and kind of figuring out like what mushroom you want to look for and if it is what type of mushroom it is, if it's parasitic, if it's mycorrhizal, or if it's saprophytic. Okay. I could have talked a lot about that, but I hope that answers.
That's great. We have a ton of people in the comments section asking you for a morel mushroom session online.
Yeah, I'll do it. I have it all ready to go. So maybe I'll put together a live session in the next couple of weeks and get that out. I even share some of my secret spots. Might have to charge for that one. No, I'm just.
Okay. We'll be sure to share that.
Yeah.
Another great question we got. Someone's wondering if you could talk about responsibly sourcing medicinal mushrooms for those who want to use them. And
it kind of cut there at the end, but I think I got the gist of it, just like ethically sourcing medicinal mushrooms, which is a really good question, or it's a really great point to bring up. Thank you, because some of these mushrooms take a long time to grow. And if you don't ethically harvest these mushrooms and kind of leave some behind, they won't be able to drop their spores and thus regenerating the forest. So I would say always leave some behind. Don't be greedy. Leave some in the forest so they can regenerate, drop their spores, and continue to grow. And also, like with the chaga mushroom, it grows in northern Michigan on birch trees. I think it grows like up in the 22nd lateral. So you want to go a little bit north to get your chaga. But the best time to harvest these is in the winter. So there's also, some can be seasonal based on when it's best to harvest them. But the main thing is to leave some mushrooms behind so they can regenerate.
We have another question from Kelsey, who's wondering if you could talk a little bit more about micro mediation at home.
Okay. Yeah, so micromediation, it's kind of I would say do your research. Make sure you're being cautious and safe if you are working with like contaminated soils to kind of know what you're working with. Maybe get the soil tested. Make sure you have hazardous or PPE, personal protective gear, if you're going to actually be going out working in contaminated soil. But how you can kind of start is obtaining a saprophytic mushroom. The oyster mushroom is a great mushroom to start with. You want to grow out that mycelium, and it's a pretty I can't really think. It's a strong mushroom, and it's not so susceptible to contamination. So it's a good for mushroom to grow for beginners. So you'll grow it out, the mycelium, and then you can like kind of mix that mycelium and some wood chips, and then let it grow out even further onto the wood chips, and then till in those wood chips into the soil, into your area of interest. And what I like to say, if you are really going to be like kind of studying this and trying to get some results and some analysis, to always have a control. So you want to have an area that is like untouched, just so you can see if this is actually working.
Let's see, lots of questions here. We're running out of time, but we can take a couple more.
Okay.
And I guess before we dive into a couple more questions, if people want to learn more, we have a couple questions wondering if you offer classes online. Where can everyone go to learn more about fungi frates?
Yeah, absolutely. I give courses all the time. And if you go to my website, which is www.fungifrates.com or .net, there's actually an area solely designated to workshops. And I'll show you the upcoming workshops that I offer. A lot of them have been canceled due to these trying times of the coronavirus, but I'll always be updating it. So yeah.
Awesome. We have a question. Someone's wondering where you would suggest to shop for lion's mane mushrooms. Someone suggested Mycophiles in Grand Rapids, but you're from the Detroit area. Do you have any suggestions?
So as just to clarify, if you're to purchase them like in a medicinal or like a supplemental form or like fresh. So my friends at the Detroit Mushroom Factory actually have lion's mane. If you're in this area, you can get fresh lion's mane. Actually, you can buy lion's mane tinctures from me. That's a medicinal supplement. I do provide that. It's on my website. That's pretty much for locally that I know of, but there's a lot of other sources. I have a friend, Olga, Smugtown Mushrooms. Hey, Olga. Shout out to Olga. She's in upstate New York, and she's Smugtown Mushrooms, and I know she offers a lot of medicinals on lion's mane as well. Or First Fungi Perfecti, which is Paul Stamets' line.
Okay, and our last question for the night. Mary is wondering if you have any tips for anyone who's sort of wanting to follow in your footsteps. If you could talk a little bit about what you studied, how you got to where you are now.
Awesome, great. That's super precious. So I don't know if you want to follow my footsteps, but no, I'm just kidding. So I started studying, I was studying like microbiology and biology in my beginning years of college. A lot of people were pre-med, so I kind of really didn't fit in. And I noticed though, there was not a lot of information on fungi at all. I'd have so many questions for my professors all the time. Like we're spending, 3 weeks on bacteria and 3 days on fungi. Like I had so many questions. I was getting very little answers. So I kind of took it into my computer. Into my own hands to kind of just start researching on my own. And I think that there's a scientist in every single one of us. Don't be afraid to go out there and do your own experiments and read scientific journals on like things that you're interested with fungi, reach out to other people in the area. And now that I have a property in East Poletown, it's really going to be dedicated to education in the mycological realm. And I want it to be an area for people to come and gather and engage in mycology together and expand citizen science. So that eventually will be up and running for everyone to come and learn. But I would just say start dabbling into books, cultivation, getting some experiments running. Yeah, kind of just doing your own research.
Great. Well, thanks again for joining us, and thanks to everyone who tuned in tonight. After this session, we'll have everything recorded if you want to look back, and we'll be sure to share more information about fungi friends.
Great. Thank you so much.
Yeah.
Awesome.